So the good news is I talked to the bartender about a 2-for-1. She's going to double the price and then give us 2 for 1. So that's a good thing. Okay, so I think where we were talking about algal biodiversity, and I thought it was important that everybody get an idea of what biodiversity was, because it's a word we use sometimes and don't define. Biodiversity basically is just the diversity of life. What does that mean? All living things comprise their biodiversity. And this includes things at the species level, so different types of organisms. It includes things at the molecular level or genetic makeup. Next week's speaker will talk about that in her work, and you've heard other people talk about using the molecular approach. And also the natural communities in which they live, so habitats, ecosystems, and so on. The number of algal species is actually unknown. There's something like 125,000 that have been described, but if you look at which ones are synonyms and so on, probably there's about 30,000 that have been legitimately described and probably are valid. So that'll give you some idea of the numbers of species that we have in terms of algae. The first person who actually classified algae at a higher level was the Irish mycologist, Wayne Henry Harvey, who divided the algae into basically 4 groups, primarily on color or pigmentation. And these were known as red algae, brown algae, and green algae because of their pigmentation that masks the chlorophyll A, which is green underneath. Depends on the type of group that you're looking at. So the red algae have different accessory pigments than do the brown algae, than do the green algae. And then the last group was diatoms, which is a microscopic group that had received a lot of attention in the 1800s, and we'll talk about them in a little bit as well. Harvey was the first person to really use biochemical criteria in plant systematics. Nowadays, we use biochemical criteria all the time. We use molecular criteria all the time. He was really the first one to start to do that, and he did it at a higher level than anybody had done before. Nowadays, the much more modern approaches, we look at molecular information, and we build. This is part of the tree of life that you can find on the internet. This is an attempt to look at the genomes or the molecular makeup of all organisms, and then show their natural relationships. And algae don't fit a single group. I highlighted some of the areas here. All green algae and all higher plants. So every plant that lives on land is up here. All animals, for example, are in this one little line here. All animals. The rest of these are where the algae are. So some of these macroscopic algae, the greens and the reds are up here, the brown algae are down here, the diatoms are down here, a lot of other algae are down lower. Most of all these other groups are all primarily microbial. So one of the things that we oftentimes forget as humans is that there's this incredible microbiota out there. And Paul Hargraves talked about that last week, and you may have heard Peter McCarthy talk in previous years about that. So algae are very heterogeneous in both in their chemistries and in a lot of other ways. I'm going to run you through a very quick overview of some of these different groups to give you an idea of the diversity that they have. The blue-green algae was a group that came after Harvey, and for a long time people would call the groups based on color. These were the blue-green algae. There was the golden-brown algae, and so on. And we now know these not to be related at all to the rest of the algae in a very close way. They're considered bacteria, specifically the cyanobacteria. And examples of these include very small coccoid cells that are photosynthetic. Prochlorococcus was not known until the mid-1980s, and we now know that maybe half of the primary productivity in the world's oceans might be due to this tiny cell. It's less than a micron, which is a millionth of a meter, and they were missed by oceanographers for a long time because they went through all the nets. We couldn't catch them. We now call these picoplankton, very tiny plankton. One of the important things about this group, as is the case for all bacteria, is that they're prokaryotic. Prokaryotic is a fundamental difference in cell type compared to the rest of the organisms which are eukaryotic, that you are most familiar with. And prokaryotic means they don't have a traditional nucleus. They have other differences in their nucleic acids, and they have very little other organizational at the cell level, so they don't have organelles. Lingbia is a filamentous alga, and we find this in the Indian River Lagoon, and we oftentimes see this floating around in the summertime. Also, people have studied these off the coast of Florida, the impact on coral reefs because they produce a lot of toxins. Valerie Paul, who's in the room tonight, who is the head of the Smithsonian down in Fort Pierce, she has done a lot of work on their chemistry. Amy Wright, who is here at 4 o'clock, also has done some work on understanding the chemistry of Lingbia. There's many, many different types of toxins and interesting natural products that Lingbia produces. Anabina is another filamentous one, and this is an example of one that has this special cell here called a heterocyst. Not all blue-green algae have this, but whenever you see that, it means that they're fixing nitrogen, that particular cell. And this is a process that goes back, as you'll see, billions of years ago when the Earth's environment was quite different. And actually, oxygen is toxic to nitrogen fixation. And what's amazing is that, in this case, there's a specialized cell that this process occurs. But there's a lot of these cyanobacteria that can do this without a specialized cell. So these very, very tiny cells, in some cases, are able to fix nitrogen, and they're able to keep the oxygen part of their metabolism separate from their nitrogen fixation part, which is very important because oxygen is toxic to nitrogen fixation. So that's very important. They're the only algae that can provide new sources of nitrogen, so they take that atmospheric nitrogen. Spirulina is, it's hard to see a little bit maybe. This is actually a spiral. It's like a coil. If you were to stretch it out, it would be like a spring. And you might know spirulina because it's grown commercially. This is actually raceways in Hawaii. And one of the main uses is actually in health tablets. And people will eat these to take some advantage of the nutrient and special ingredients that some of these algae have. Historically, this was eaten as food in certain parts of the world and became more of a health item in our part of the world. Diatoms, I mentioned them earlier as being one of the original groupings in the 1800s. And diatoms are interesting because their taxonomy is all based on the dead cell. And the dead cell is all this silicon or glass cell wall. So think of these as little petri dishes. You know what a petri dish is. And there is a top part of the dish and a bottom part of the dish. And the diatoms are sort of put together in the same way. And diatoms come from the Greek word cut in two. And that refers to these two different parts of the cell, or valves, if you will. Diatoms are very, very important ecologically. They're important as aquaculture feeds. Skeletonema is a well-known diatom that lives in coastal and estuarine waters. It's in the Indian River Lagoon. You can find it throughout the world, up and down the East Coast. There's also some centric diatoms, which have these really nice markings. And the whole taxonomy is based on the details of those markings. In the 1800s, this was actually sort of a hobby where people would place these under a microscope slide and make designs, and then they would send them, out to people as a gift. Some of them have special adaptations like spines. This is Catastrophes. There's many species of Catastrophes in our area. And the spines can be useful for flotation and also to discourage grazing by zooplankton. Dinoflagellates. And it is dinoflagellates, not dinoflagellates, although you hear most people say dinoflagellates. Dinoflagellates comes from the Greek word meaning whirling, and flagellum, which is a Latin word for whip. So they're whirling whips. Flagella are like little whip-like appendages that look something like that. Dinosaurs, as you know, are terrible lizards. That's what that word means. And so if you said that dinoflagellate, then you would mean terrible flagellates. And in fact, this is a whirling flagellate. Okay. And you may know these as red tides, because some of them produce red tides. And one of them in particular produces the Florida red tide, which is this guy here, Carina brevis. And that's actually named, the genus name is actually named for Karen Steidinger, who has worked for many years over in St. Petersburg on the Florida red tide. Another interesting one that we find in the Indian River Lagoon is this one here, Pyridinium bahamensi. Some of the strains are toxic, and in particular, they produce a saxitoxin, which is a neurotoxin. very potentially deadly. And a few years ago, there was incidents where this had been concentrated up in the food chain into pufferfish. People who were eating pufferfish in the Inner Lagoon got very ill. And so we now have since then banned pufferfish in the Inner Lagoon because of this potential problem. But dinoflagellates are not all bad. This is Symbiodinium, and you'll hear about corals next week, and you'll hear probably a little bit about coral bleaching, and you've heard about coral bleaching in some of our other lectures. Symbiodinium is a non-flagellated dinoflagellate that lives inside the corals as a symbiont. And its relationship with the coral is very important to the health of the coral. And you'll hear more about that next week. But this is why when you swim over a coral reef, they're golden brown. Okay, it's because of the color of the zooxanthellae was the earlier name for this particular group. And zooxanthellae is still sort of generically used to mean those symbiotic organisms. Coccolithophorids, another group of microscopic algae. They're little green guys. They got two flagellates, but they also have something in between that's called a haptonema. And the group, taxonomically, the haptophytes, was named because of the haptonema. Haptonema refers to this little extra flagellum-like structure, and it comes from the Greek meaning touch or fasten in a thread. And what this is believed to be, the function of this is believed to be more in terms of particles, ingesting particles, or also sensing the bottom, things like that. What's amazing about coccolithophorids is they produce coccoliths. That's what coccolithophorid means, bearing coccoliths. And these are all of these calcareous scales that grow external to the cell. So these individual unicellular organisms produce this incredible ornamentation. And when the cells die, they oftentimes will settle to the bottom of the ocean. This is more of an oceanic type of group. And you may know the White Cliffs of Dover. This is a geological accumulation of coccolithophorids. This is all of these guys, fossilized. Okay, so that's a rather impressive thing to see. Macroscopic algae now, the red, brown, and green algae. The red algae are named because of their rose and plant. Whenever you see phyta, it always means plant. Rhoda means rose or red. So in the Inner Lagoon, we'll talk a little bit later about Gracilaria t. That's probably the most abundant one in the Inner Lagoon. Maybe you walk along the beach or you snorkel out to the worm reef, you might see this beautiful alga, Botryopladia, which has these vesicles that look like little pearls underwater. They're important red algae that are calcified, and these are important in coral reef. So this is Neogonulithon, and think of these as sort of the organisms that cement together the different parts of the reef. So it's not just coral that produce that structure. There's a lot of coral and algae that also contribute to it. And these algae are also very important for settlement of coral larvae. They will preferentially settle when there's an alga like this out there. So they're very important for coral reefs. And here's another one that is more upright. We call that an articulated coral. That's Jania. We actually have Jania locally in the Inner Lagoon. It's quite an interesting story, which I won't go into now. A lot of red algae are economically important. Some that come to mind to me right away is Porphyra. There's lots of species of Porphyra, but the really good ones are cultivated in Japan, and they make these dried sheets of nori. And you probably know nori because it's the wrapper on it's the outer part of your sushi. So if you like sushi, what you're eating is various things that are wrapped with nori. Okay? And they're grown, they've been grown since the 1600s on basically ropes and nets like this. And again, there's quite an interesting story as to how that all happened. Another one is Eucheuma. This is grown more for a commercial product. There's a whole category of compounds that come from algae called phycocolloids. And Eucheuma is a source of carrageenan. Carrageenan is one of the key ingredients in ice cream, for example. And these are grown most predominantly now in the Philippines. And they're often harvested by fishermen, and oftentimes it's an alternative source of revenue for them. And again, they would be cultivated on rafts or lines of some sort. Gracilaria is another one that has been grown commercially in various places in the world. In the 80s, there was increase in gracilaria production in Chile, and then more recently it's been in Indonesia. And again, here's an example of what it looks like in Indonesia. Again, this is the type of thing where it's open water, usually very shallow coastal waters. It's labor intensive, but the labor source is very cheap. Oftentimes, again, it's secondary revenue. Oftentimes, it's like family of fishermen that will do the work, and they sell it to the phycoalgae companies. Let's talk about green algae a little bit. Green algae means green plant, and most green algae are actually tiny, microscopic forms. Most of them are freshwater forms. This is Chlamydomonas. Chlamydomonas is like a white rat in the phycology world. It's used tremendously for plant physiology studies because it being a green alga, it has the exact same pigment and the exact same biochemistry as do higher plants. So it's a lot easier to do physiological work with a culture of Chlamydomonas than it is to say do it with a larger tree or a corn plant and things like that. So a lot of basic physiology that we know about plants that have been modified for, say, agriculture derives from work on Chlamydomonas. So it's quite an important plant. I was thinking when I was putting this talk together, what was the first time I ever saw an alga? I decided it was in high school when I was taking my biology class, and I think it was probably Spirogyra. I've always liked Spirogyra. I don't know if that's the reason or not, but it has a spiral chloroplast. That's where all the pigment is, and so it's very easy to tell the genus of that particular group. So maybe you've seen that in your biology classes. And again, as I mentioned earlier, these are all ancestors to land plants. So the evolution of terrestrial plants came from green algae, and that was a very important event indeed on the planet. I'll talk about in a little bit. So some of the bigger forms, and some of the ones you may have seen, you may know sea lettuce, called Lactuca, and that is grown locally in the lagoon, and I'll talk a little bit more about it later. The plant that I chose to work on for my PhD work in Rhode Island was this one, Codium fragile, which actually was an invasive species, and it was back when we didn't know much about invasive species and what was important about their characteristics that made them invasive. And this is one of the really first algae that we knew to be an invader throughout the world. And it was first introduced in Long Island Sound and then spread up and down the coast. And one of its common names is oyster thief, because it would grow on oysters, and after it got really big and buoyant, it would just pop the oyster off the bottom and float the oyster out to sea onto a beach. More recently, this is an invasive species in Florida. Maybe you heard Brian LaPointe lecture about Chloropa brachypus. This is a species that is known primarily from the Pacific. We don't really know how it got here off the coast of Florida. Probably aquarium trade, but we don't really know that for sure. And in between the time this appeared in 2002 or so, in the early 90s, we had quite a large amount of Codium ismocladum, a different Codium species growing in that same area. So Brian and I were both working on that, and then he's basically done it for 20 years since then. There's also calcified green algae, such as Halimeda. This is very common in the Florida Keys. We have some locally, but it's much more dominant in the tropics. Halimeda is important because if you look at the sands on the beaches in, say, Florida and the Caribbean, most of that's calcified material that's been produced by organisms that has been ground up, and the highest percentage of that comes from this alga, Halimeda, this genus. If you go to a place like Florida Bay, where there's a lot of fine sediments, more mud-like, they are actually derived from a lot of the calcified algae, such as Penicillus. So some of these algae are just very important geologically and also in terms of sediment cycles. Let's talk briefly about the brown algae. Locally, the ones we probably know the most about, the ones that you probably have seen, is the sargassum that's cast ashore, or maybe you go out fishing and you know that if you go out and find the weed lines, the sargassum weed lines, you know that's where the fish are. And maybe you've actually gone to the Sargasso Sea. I did that once. That was a big treat for me. The Sargasso Sea is actually named for the plant Sargassum, for the alga Sargassum, which was named by the Portuguese sailors after a species of rock rose called the same name, Sargasso, in Portuguese. The story goes that Christopher Columbus in 1492 was going across, and the troops were getting a little restless. They wanted to mutiny, and they were in the Sargasso Sea, and they started to see patches of this. They named it for something that reminded them of home, and I guess they continued on to discover what became the New World. By the way, in 1493, the following year, I believe, is when, if you look through Columbus's journals, he mistakenly called the manatees mermaids. So that's how that whole thing started too. So he had quite a history of adventures when he was here. In the Indian Lagoon, the largest seaweed we have is a sargassum philopendulum, and it grows attached, and it grows quite locally here to Harbor Branch. If you go to the Florida Keys, you oftentimes hear algae overgrowing the reef. Generally, we're talking about Halimeda, but also this brown seaweed, Dictyota. It's very, very common, very important. Also has very interesting chemistry, which Al Paul can tell you about. A large group of brown algae that we do not have here is the kelps, but you guys have been various places in your life, and I'm sure that you have seen kelps if you are in more temperate waters. Laminaria is the genus name. That is found in New England and actually all throughout the northern part of the world. You may know Macrocystis. It grows in various places in the world, but off California, it can be 200 feet long, 65 meters. Largest plant, largest alga in the world. Maybe you've gone to the Monterey Bay Aquarium. If you haven't, you should, because this is the best place to see algae where you don't have to get wet. And they do a wonderful job not only displaying natural populations of Macrocystis, but they probably have 60 or 100 species that you can go around and look at on the algae. So I love the Monterey Bay Aquarium. Maybe you know that for many years until very recently, there was a whole industry off of California. This is a boat harvesting Macrocystis. And that was harvested for a number of reasons over the years, most of the time for alginates. Alginate is another phycocolloid. Alginate has a lot of uses. It's, for example, in a lot of food products. Maybe you know alginates because if you ever had a dental impression and they threw something in your mouth, out came a mold, voila, that was alginate. Okay. In Japan and China in particular, they grow Laminaria japonica, which the name there is kombu. And this is the leading fisheries product really in the world. And this is primarily totally for food. And there's a whole other side of seaweeds that we don't see in the United States. And that has to do with the food side of it for human food consumption. That's mostly entirely really in the Orient. Next thing I want to point out is that algae have been here a really long time. And I want to take you back through the fossil record. The very oldest fossils that we know on Earth, from 3.9 billion years ago, were probably the first cyanobacteria. About 500 million years later, so it took a while, we have fossils of the first oxygen-producing cyanobacteria. I said how important that was because it was a very different environment. There was no oxygen in the air. It was a very reducing type of place. Take half that time from 3.4 till now, and it took another 1.8 billion years before the first multicellular algae were found in the fossil. There's evidence in the fossil record. These are all red algae and green algae, so they are the oldest macroscopic algae. Much more recently, almost 500 million years ago, we started to see the other group of brown algae. Remember I said earlier that molecularly we now know they're very, very different than the other two. And that was a much more recent event. And then about 360 million years ago, we got those copperlithophorids, and I mentioned the white cliffs of Dover. They're those organisms. And about 200 million years ago, the first dinoflagellates, and then much, much more recently, the first diatoms. So this has taken quite a long time to get this great diversity of algae. And this is just a very quick plot through time. It's like 4.5 million years ago. So the first algae were out here, and you can see that this is oxygen relative to now. So 100% here is what our oxygen level is now. And you can see that for most of the life on Earth history, the oxygen levels are much, much lower. And then when the cyanobacteria started to produce oxygen, at some point they reached some threshold, and it really started to spike. Land plants, so these little simple algae got about half or 60% of the oxygen level up. And then when land plants formed on land, that really kicked up the oxygen level, and now it's been stable for several hundred millions of years. So it's kind of interesting. But it shows the relationship of our global environment to these organisms, and the fact that something that happened maybe a billion years before led to conditions that allowed some of these other things like multicellular organisms, which without an oxygen environment could not exist. So this is actually kind of really important, I think, to hook the fossil record with what happened with oxygen levels. Algae are important for many reasons. I've listed them here, and I've mentioned a lot of these in my examples earlier, so I'm not going to go through these in great detail. I'm going to pick one later. Also, I mentioned a lot of the economical uses of algae, so I'm not going to go through them. But I will mention two others. One is fisheries. There's a very important relationship of algal habitat and algal food supply to fisheries. And you know, think of the macrocystis forest off of California and the relationship it has to the fish that live in that habitat. Another thing I think is again using the kelp idea, recreation. I mean, sometimes these are just great places to go. I mean, if you've never gone scuba diving in a giant kelp forest, you should do that if you can. It's really quite cool. And it's hard to put dollars on some of those. So let me now talk about a little bit about some of the reasons why algae and plants in general are different than animals, and why they do things that we may not think about right away. First of all, here's a little chemistry, little equation for photosynthesis. This is really one of the most important things that happen on Earth. Plants, including algae, in the presence of light, so photons of energy from the sun, and the presence of that master pigment, chlorophyll A, can fix carbon dioxide from the atmosphere, from the water, and produce sugar. This is glucose. We produce sugar, and then give off oxygen. So they're fixing carbon dioxide, they're giving off oxygen, they're making food. That food, they can take up, sorry, let me just mention one other thing. I may talk about productivity when I talk about this process. That's the rate at which organic matter is produced. I might talk about production, which is the total amount of organic matter that's produced. So productivity is more a rate per unit time per unit area. Production is more like the total amount. And algae, just like plants, can take up inorganic nutrients directly from the environment. So what does that mean? Well, they can take up nitrogen from the environment as ammonium or nitrate, and they can take a sugar molecule, they can add an ammonium ion to it, they can make amino acids, and then they put the amino acids together to make the proteins. So they do this all from scratch, so to speak. They take the energy from the sun, they take the inorganic elements from their environment, and they make biomass. They make living material. Animals require that to be existing already. So animals eat things. Okay, so animals don't have that capability to directly make energy from the sun or to take up inorganic elements. That's what plants do, and algae. So one of the recurring themes is that energy flows. So energy from the sun going into that primary productivity level, such as algae, being consumed, say if it's a phytoplankton, being consumed by a zooplankton, being consumed by a small fish, being consumed by a bigger fish. At each step, there's a loss of energy. Energy's flowing through the system, but nutrients are cycling. So when those plants take up the nutrients, when they die, they're going to release the nutrients. The animals will take up the plants and they'll excrete nutrients. So nutrients are constantly cycling in the system. And I'll bring these points up later. So I want to give you one concrete example. The inspiration for this was, I was trying to think, well, maybe how could I explain some of these terms, some of these ideas to you all, to understand how little we really understand the importance of algae. And a couple years ago, I had a colleague at a meeting give a talk, and he presented the basic concept here that I'm presenting. And his name is Bob Anderson. And I talked to Bob recently electronically, and he said, yeah, go for it. So I'm presenting some of this based on his inspiration to me. And it basically starts off with the premise of how many cells of phytoplankton, these little algae, are in the oceans at any one time. Now I sort of tried to ground truth this yesterday, and I found numbers all over from 10 to the 23rd to 10 to the 29th cells in the oceans at any one time. Those are huge numbers. I kind of took the average, or the most commonly cited numbers, and that turns out to be about 1 times 10 to the 24 cells. I don't know that we have a name for how many powers that is. It's long past any zillions of numbers that I know. But 10 to 24 cells. They're very tiny, though. They're only 2 microns, or micrometers, average diameter, because most of them are small phytoplankton in the ocean. And that's about 0.0008 of an inch. So really small. These are basically bactericides, or a little bit larger on average. So the question is, if you've got 1 times 10 to the 24 cells, they're all 2 microns wide on average. If you put them end to end, how long of a line would that be? Anybody want to guess? I know you got an answer. All right, I'll tell you. It would go from the Earth to the Moon and back. Pretty incredible, right? But it doesn't do that once. It doesn't do that twice. It doesn't do it 3 times. It does it 1.5 billion times. Now, that's really hard to imagine. I can't imagine it. It's hard to imagine the magnitude of this number. It's hard for me to imagine how small these cells are. It's hard to imagine how many times back and forth from here to the moon. So, maybe we can do this a different way. We can arrange the cells into 2 dimensions. And so we just pack the cells together like that. And we can make this, say, 12 inches wide and 3 inches deep. So it's a piece of wood. Think of it as a piece of wood, 12 inches long, 3 inches deep, something like this. How long would that piece of wood be? How long would that piece of wood, how long would that piece of algae be? A plank of algae. Anybody want to guess? It would go from here to the moon. So imagine that. So you see, we could actually build this, and we could actually do the walk to the moon. But that's incredible when you think about that. Now, here's what's even more incredible in my mind, is that on average, phytoplankton are dividing once per day. So that means they're doubling every day. So what does that mean? That means you'd have 2 boards tomorrow. Okay? But we don't have 2 boards tomorrow. Why is that? Because one's being eaten every day. It's eaten every day by protozoa, little animals, microscopic animals, zooplankton, fishes. You know, sometimes whales will feed on phytoplankton. So this daily board, if you will, of phytoplankton is what feeds the ocean. And if you eat seafood, or if you still get some revenue from the sea because of marine organisms, you're depending on that board of phytoplankton. So I thought that was a way to try to relate it. Point that algae provide food for animals in the estuaries and the oceans. And in so doing, I showed you the equation that they fix carbon dioxide and they produce oxygen. And I showed you that through geological time. That's why we got an oxygen environment instead of a toxic atmosphere that none of us could live in. But let's talk about today. Okay, every time I take a breath, 50%, half of them, that one's from the land. And my other breath, out of those 2, the other 50% is from the oceans, from the algae. Okay. Second one was a little saltier, by the way. That's amazing when you think about it. You know, think about that. Every time you breathe, where'd that oxygen come from? It came from the plants. Another way to do that, of course, would be you could do all your 12 hours of breathing, you know, on the terrestrial plants, and then you'd skip the algae for 12 hours, so you couldn't breathe for 12 hours. That I didn't want to demonstrate that. So the oxygen every breath we breathe comes from algae. So therefore, without algae, we would not be here, even in terms of the long scale, evolutionary-wise, or in terms of our more daily life. So I want to phase into the second part of my talk a little bit about some research stuff. Indian River Lagoon, a lot of you know about this. I've talked a little bit about the lagoon in the past, but you know, a lot of you live right here, and you know about the Indian River Lagoon. The Indian River Lagoon is really special. It's one of the longest lagoons in the continental United States. It's about 40% of the east coast of Florida. The Indian River Lagoon is often called the most biologically diverse estuary in North America. This is due to its large numbers of species, a favorable climate, and we get species that come from more tropical waters to the south and more temperate waters to the north. And the lagoon is just an incredible fertile mixing bowl of organisms, of life. You also know that we're living in an area that is rapidly being urbanized, so it's a challenge to have such a productive natural system that we may potentially be impacting. If you care more about the dollars, the economic impact of the Indian River Lagoon in the last year or so was calculated to be more than $3.5 billion per year for Florida. So that's every year, $3.5 billion. In the early 90s, the water management districts and other agencies decided that a high priority for maintaining the biodiversity in the Indian River Lagoon is to keep it a seagrass-based ecosystem. And why is that? Bottom line, those of you who heard me talk before, more seagrass you have, the more fish you have, because it's great fish habitat and it's great for major invertebrates as well. So I was always interested when I first came to Harbor Branch how little research was done locally on the Indian River Lagoon marine algae, and a lot of the literature was old, outdated. Honestly, it was quite a challenge to work on the macroalgae of the Indian River Lagoon. But some of you may have heard a talk I gave in 2008, I think, with Mark and Diane Littler. They were the first authors. Diane was the first author, then Mark and myself, on this book, Submerged Plants of the Indian River Lagoon. And it's probably one of the funnest things I've ever done in phycology because of them. And to me, this just says the tip of the iceberg about biodiversity and about algal biodiversity. We recorded 229 species of marine algae, which was a lot more than anybody thought was in the Indian River Lagoon. But what really strikes me as amazing was that more than half of those, 57%, were new records for the lagoon. Now, this lagoon is right outside our doorway. The average depth is 5 feet, 1.5 meters. We've lived here for a long time, and yet in 2008 we can go out and decide that we have all these new records for the lagoon. 20% were new records for Florida, and 1% was new records for the Western Atlantic. So to me, that just tells me, what an exciting place the lagoon is. If you're interested in the breakdown, the flora, so to speak, is dominated by red algae, 112 species, and then green algae, and then brown algae. That's very typical of warm water and tropical waters in the world. Most of the algae, macroalgae in the lagoon, is so-called drift algae. And that's because there really isn't much hard substrate in the lagoon. As you know, it's soft sediment. And there's not a lot of places for the algae to attach, which is what you see when they're rocky areas. And so the algae actually does move around based on winds and currents. And it's actually very hard to know how much of it is being produced in one location versus how much is drifting in from other locations. This is a picture of Sherry Reed. Sherry Reed was at 4 o'clock lecture, and she was a huge help in this project. And just really loved working with her too. So I got a picture of Sherry. And what we were really interested in is being able to identify some of these drift algae, which before this was incredibly hard to identify. If you go back to the literature, you don't really know what anybody is talking about when they refer to some of the algae that's been reported in the lagoon. So we now have this book, and many of you have seen it, and hopefully I think it's going to really trigger a lot of additional work on the macroalgal community. It's helped me because I'm very interested in things like, well, how much macroalgae is actually in the Indian River Lagoon? So I can tell you that I know it's highly variable in time and space. And I have consulted with Lori Morris at the St. Johns River Water Management District. Lori and I worked together periodically over the years. And then again, another wonderful person who works in the lagoon. And she has done some estimates with some of her colleagues that if you take the Indian River Lagoon segments south to Indian River County, so not the whole lagoon, and she did this for 2 years, she got about 70, let's call it 80,000 metric tons. That's a lot of algae. I did an extrapolation, so if it's wrong, it's all on my shoulders. But taking the area of that study and the entire area of the Indian River Lagoon system, my estimate is that there's probably 170,000 metric tons of algae in the lagoon. That's quite a lot. So one of the obvious questions is, how does it tie into, say, nutrient loading into the lagoon? And in total, the lagoon receives quite a lot of nitrogen and phosphorus. Most of this comes in from discharges from freshwater sources, the canals that we all know about and some of us live on. And in this paper, which again is hooked to the Water Management District, there was this comment about that the current rate of nutrient loading is causing a shift in the primary producers of the Indian River Lagoon from macrophyte, meaning seagrasses, to phytoplankton and algal-based system. And so there's some concern raised about that. Now, how true that is, I don't really know, but it's certainly a question that I'd like to answer. And in particular, what I'm interested in is the growth of macroalgae in the Indian River Lagoon limited by nitrogen. And does macroalgae growth in the lagoon then respond to increases in water column nitrogen? So I work mostly in the local area from Vero Beach south to Fort Pierce and the Fort Pierce Inlets down here. And we've had a number of stations in that area for a couple of years. And if you're interested in the overall patterns from those discharge points in Vero Beach as you go from north to south, this is now nitrogen. You can see it decreases through time. And the first year was a wet year, and we had a lot of rain and a lot of discharges. The second year was a beginning of a drought year. And you can see that it does matter how much nutrients are discharged depending on the weather and the discharge rate. And you can see that for phosphorus, perhaps it's even more dramatic. So this is nitrogen, that's phosphorus. Those are the two main nutrients when we talk about algal growth. Over the years, I've been very interested in using algae like biomonitors and looking at the tissue nutrients. And this is productivity, so that's the rate of primary production, grams per meter squared per day. And you can see this is internal nitrogen, what's actually in the plants. Initially, when you increase the nitrogen in the plants, it increases its growth rate really fast, very explosively, and then it levels off because it's got as much as it needs. So think of this like when you fertilize your lawn. You know that you need to fertilize a certain amount. If you add too much fertilizer, it doesn't do any good. It's actually waste, and actually it's probably not an environmentally good thing to do. So I've used that over the years, and I've done some experiments like this one, where I've looked at the productivity of things growing here in the Inner Lagoon, and they grow very fast. They start to slow down, and you look at the tissue content of nitrogen, you see that's because the nitrogen level now gets below this magic 2% number, this critical level, and it starts to go slower and slower. If you add a little spike of nitrogen, that's what this arrow is, you can see that the internal nitrogen very rapidly increases, as does growth rate. And you can repeat this on, and if you do a control setup where you add no nitrogen and just let it grow, you can see it just sort of slowly continues to grow slower and slower and slower, and eventually just sort of zero out. So we've looked at this through the year, and we see this. This is that 2% number for tissue culture. If it's above this number, that means it has plenty of nitrogen. If it's below 2% level, it means it doesn't have enough nitrogen. It's hungry for nitrogen, starved, if you will. So you can see that in the dry season, that's when you have the lowest tissue nutrients. That's when nitrogen is most likely to be limited. And then in the wet season, after we've had extensive discharges, that's when you have the maximum amount of nitrogen. So it does seem that it tracks it very well. North to south, this is what it looks like. So this is Vero Beach up here. This is where Harbor Branch is. So Harbor Branch is right about that magic 2% number. And then closer to the inlet, you can see there's probably nitrogen limitation. So the answer to the question is it depends where you are and when you're there. So from north to south, nutrients decrease. Pulses of inorganic nitrogen are usually associated with the initial onset of the wet season. And if we have tropical storms or hurricanes or any kind of major discharge, we see these pulses of inorganic nitrogen. The algae are able to take them up very rapidly, and the tissue nitrogen reflects what's in the environment. And we've done studies that we have looked at the growth rate as a function of the nitrogen content, and we see the relationship that I mentioned earlier. So nitrogen limitation does occur both spatially and temporally in the segment of the Inner Lagoon. And so what I think this means is that there's a lot of proposals out there to reduce the amount of freshwater discharges in the lagoon to reduce nutrient loading. There's going to be an EPA meeting here at the end of the month where they're going to try to set targets for the Inner Lagoon. That'll be very interesting. And I think that if we are able to reduce those nitrogen loadings, we should see a decline in macroalgal productivity and abundance, and it should lead to reduced competition of macroalgae with the seagrasses in the lagoon, so the concern that was raised earlier. And then very briefly, I want to talk about algae for biofuel, because actually that's why I first came to Harbor Branch. I didn't mention that. But I was going to solve the world's energy crisis a while back. How are we doing? Still working, okay. Remember that formula for photosynthesis, very important. Plants have this incredible capability, and algae has this incredible capability to take light from the sun and make biomass. And remember fossil fuels, that's what we all use. To get here today, how'd you get here? You use fossil fuel, right? That's how I got here. Fossil fuels are formed by anaerobic decomposition of organisms, especially phytoplankton and zooplankton, usually in deep anoxic seas or lakes, and this was done millions of years ago. Fossil fuels are not renewable unless you think that you can wait millions of years for it to happen again. So they're not renewable. This is addressed to the baby boomers in the crowd, especially me. I'm a baby boomer. I find this a sobering statistic. Maybe I shouldn't do that St. Patrick's Night, right? Here you go, though. In one generation, if you were born in 1950, and the average life expectancy for a baby boomer in 1950 is 75 years to 2025. During that period of your one life, that one generation, 70% of all U.S. oil will have been consumed. You can see it was much lower before that generation, and it's going to be much lower in the future. That's amazing. So basically, one generation of Americans took and used 70% of all the oil that we have. Okay, that's incredible. So, could algae be used for biofuel production now? Don't wait a million years. If the algae can do this marvelous thing, why can't we capitalize on it now? And in fact, I said this is why I came to Harvard Branch in the first place, and I came because John Ryder had started some projects here in Florida at Harvard Branch. Previously, in 1969, he started what he called at the time a tertiary, marine aquaculture tertiary sewage system complex. And the whole idea here was to take raw sewage or secondarily treated sewage, grow phytoplankton. Algae love nutrients. I've already talked to you about that. The best thing you can do with phytoplankton is feed it to a filter feeder, like a shellfish, like a good old plant. And what he found was that the phytoplankton could take up a lot of the nutrients. That was a good thing, because back then we were still discharging sewage out quite a lot to the marine environment, much more so than we are now. And he found that the shellfish readily consumed and grew quite well on these phytoplankton. But the problem was they're animals, so they excreted all their waste. Remember, nutrient cycle, right? Energy flows, nutrient cycle. So they excreted everything back out, and he started to use seaweeds as the last step. He called it a polishing step. And the idea here was to strip out all of the nutrients from the shellfish. Well, it didn't take long for people to think, hey, the seaweeds are growing incredibly fast. We had no idea seaweeds could grow this fast. They're taking up all of the nutrients. Why bother with all the in-between steps? Can't we just go from sewage to seaweed? And in fact, that's one of the first things we did here at Harvard Branch. And the answer is yes. And this is a time when we had just gone through oil embargoes in the 70s. We had gas rationing, so you'd either get your gas on an odd day or even day, depending on your birthday, things like that. And there was quite a demand in the country to do something about renewable energy and finding alternative sources to fossil fuels. And that led to a series of years, about a dozen years, where we were funded by a bunch of agencies here at Harvard Branch in our old Aquaculture North complex to grow seaweeds for biofuel experimentation. We focused, after a lot of species screening, we focused on two groups, Gracilaria, multiple species, and also sea lettuce, all both of them are very productive. These are much higher yields than you find for terrestrial crops, for example. They grow year-round. A lot of terrestrial crops are seasonal. They only grow in summer or a certain part of the year. And some of this was really the first time that we understood that macroalgae could grow really fast when conditions were right. We did a lot of work on things like light and temperature and nutrients and carbon dioxide, trying to determine how we can maximize or optimize growth. This is a slide that just shows the difference between night and day. This is dark versus light versus how fast they can take up ammonia, the nutrient. And what that shows you is that's a lot more efficiently done in the light than it is at dark. So if you're interested in nutrient recycling efficiency, you're going to fertilize in the light and not overnight. I actually did my own digesters. These are anaerobic digesters. Methane gas was the product, which is the major ingredient in natural gas. About 60% of what comes off the digester is, in fact, methane that you can use directly. And again, keeping in my theme of nutrient recycling, one of the things I was really interested in was then taking the waste, the residues left behind this digestion, which is liquid and some solids, and then recycling it back to the Gracilaria cultures. And we basically closed the loop here. So we could grow the Gracilaria, put it in a digester, tap off the energy, methane, and then put back the nutrients and keep using the same nutrients over and over and over again. And that actually was quite a lot of fun to do that. So again, energy flows one way, but nutrients cycle. Since then, we've used seaweeds for a lot of other purposes. We have some people that we work with that use seaweeds, Gracilaria, some of the reds, to feed to Ecclesia for neurobiological research, because the neurons of Ecclesia are great models for our neurons. So it's quite a lab rat. And we've used this for diets in conch, and you've heard some presentations in this series about conch. We've used it in diets for ornamental fish, such as these, clownfish. And we actually use seaweeds as they are for home aquaria use, because it's a pleasant, aesthetic thing to have in an aquarium. And also, it's very nutritious for the herbivores that are in the tank to eat seaweeds. So why did all these efforts on biofuels stop more than 20 years ago? Well, bottom line is always money. Unfavorable economics. Back then, I calculated that growing seaweeds for this purpose was worth something like a penny a pound. And it's worth a lot more for food or for aquarium use, all kinds of other things. So energy prices were low in the United States. There was unfavorable political climate, limited interest in renewable energy. There was sort of a resistance to try something new and different. I think this was a lack of vision, because I think, just showing you that one graph about the baby boomer image I portrayed there. I mean, at that point, we already knew where the amount of fossil fuel was headed. So we knew it was a long-term problem. It requires long-term solutions. It's not something you're going to fix in a year or two. So how has it changed since then? Well, this is prices of oil, barrel prices. And the work I'm talking to you back then was back here, when we had these oil embargoes back here. And you can see the price now is still very historically low back then. You know that over a couple of years ago, we had this huge spike in prices, and then it fell back. And it fell back because of primarily a global recession. The important thing here is the increase we saw in the last 10 years is due primarily to a sharp increase in demand. It's not the politics of the 70s. It's not an interruption in the supply. It's primarily driven by increase in demand. It's because there are other countries in the world, specifically China and India, that have greatly increased their fossil fuel consumption because they want to do the same kind of things that we do. And there's a tremendous amount of cars and things that are in those countries now that 10 years ago weren't there. So it's just a global problem now. World oil reserves have peaked. This has peaked really in the late 80s. This appearance of an increase here is just because another category was added, which is Canadian oil sands. There's some serious problems with Canadian oil sands. You can see the 60 Minutes episode that was on last year if you want to know more about that. But you can add them in or not. The point is that they're not increasing. They're very stable. So it's not like we're going to find a lot of new oil. Believe me, people have been looking for a long time. Our use of oil exceeds our discovery of oil. So obviously at some point we're going to severely decline in how much we have. And the best projections I have is that it's going to be about 40 years of reserves at the current rates of production and discovery. That could change. It could be less than 40 years. And you also know as you start to run low, as you found out a couple years ago, oil prices can change very rapidly, and that may very well happen someday. You've seen this graph multiple times in the lecture series by different speakers talking about the climate change and how it's all tied to carbon dioxide increase in the atmosphere. And that, again, is tied to our use of fossil fuels. So I'm not going to belabor that one because you've seen that one a lot over the last 2 years. Algae have many advantages of renewable resources compared to agricultural crops. They have higher yields, higher rates of growth on a per unit and area basis. They grow year round. They are readily convertible to methane, as I showed you earlier. And they also are a good substrate for cellulosic ethanol, which is one of the big interests that has been used for some of the agricultural crops. What I like best about it is that it doesn't impact food prices and availability. We're not going to take productive agricultural land that really should be used for food and convert it to energy. And there's recent work that shows when you do that, actually, you have a net loss overall because you have much elevated greenhouse effect because you're disrupting the natural environment if you start to do new agricultural lands and things like that. So you can read about that. It's been in a lot of places like Time and National Geographic if you want to know the story on agriculture and ethanol. The renewed interest in algae is primarily microalgae, and what I want to point out is that for many years, 20 years almost, there was a study by the Department of Energy on renewable fuels from microalgae, and we were involved in this early on at Harbor Branch. Bottom line, it was not economically viable when they closed it in 1996. The current interest in research and development is primarily biodiesel and liquid biofuels. That's why the interest in microalgae. Here's a quick picture of somebody who has a system that's a lot more high-tech. It's in a controlled greenhouse environment. The idea here is to you grow a lot of the algae in small volumes to maximize light energy transformation. And this is an artistic rendition of what a large biofuel farm, say in a desert in the Southwest, could look like. Why microalgae? Because they produce oil directly. This is Botryococcus, and you can see little beads of oil being produced. And this is diatom, cyclotella, and you can see oil droplets here and external to the cell. So this is why microalgae have become quite popular. Now, the reality is that oftentimes the algae will produce more oils when they are growing less, so there's quite a trade-off there. I started really taking this seriously again. I thought this would happen a lot sooner than it did, but I think more seriously when I started to read about ExxonMobil, for example, putting $600 million into liquid transportation fuels from algae, and they had bought Craig Ventnor, who is a genomics pioneer who did the human genome, among other things. And he's also documented incredible biodiversity using molecular techniques in the world's oceans. And so then one day I was home, and I was getting up to take a break at a commercial on a Thursday night, NBC prime time, and I was walking out the room, and I heard this guy in the commercial say, "I've been growing algae for 35 years. I stopped dead in my tracks and said, "No, I can't believe this is on TV." And so let's dim the lights and we'll see what I saw.
I've been growing algae for 35 years. Most people try to get rid of algae, we're trying to grow it. The algae are very beautiful. They come in blue or red, golden, green. Algae could be converted into biofuels that we could someday run our cars on. Using algae to form biofuels, we're not competing with the food supply. They absorb CO2, so they help solve the greenhouse problem as well. We're making a big commitment to finding out just how much algae can help to meet the fuel demands of the world.
And I just could not believe that. And Joe Weissman, who is in there, some of the folks in the room will recognize him. He worked at Harbor Branch, was based here at Harbor Branch for a long time, growing phytoplankton for clams. And he was here for many years. And now he's involved in that project, which is based largely in California. I think biofuels is part of the solution to the need for alternative energy. I think there's, my preference is always going to be for things like wind and solar, which make quite a lot of sense in certain parts of the country. And also, you know that FAU is doing some work in the Gulf Stream area, Florida Current, with turbines to try to tap in some of the energy that flows through that incredibly strong current. So I think this is something that's going to happen, and I think that biofuels will be part of it. I think that macroscopic algae, seaweeds, that I'm interested in, make a lot of sense for being part of that mix, especially in places like Florida where we have year-round growing conditions. And I always appreciate when I see other folks, such as my colleague Terry Chopin, who was a postdoc here about 20 years ago, who's working on something called integrated multi-trophic aquaculture, which basically is a fancy way of saying he is growing multiple organisms that have different functions. This is out in the water, and there's a salmon farm here, cages of salmon. And the salmon, just like I said earlier, for any animal, are going to produce a lot of waste, a lot of nutrients. So they have mussels that take up the particulate matter, and then they have algae, in this case kelp, laminaria, which assimilate the inorganic nutrients. And one of the things that Terry's really done well with is he realizes that this is more than just science. It's sociology, it's politics, it's getting the people in the community behind this, and he really has built quite a very interdisciplinary team. I think you're going to see more of this in the future. I think some of the students in the room will be part of this. Okay, so bottom line then, why are algae underappreciated? Well, I found this quote a number of years ago when I first started teaching a marine botany class, and it goes like this: plants are the most important, least understood, and most taken for granted of all living things. And I think that's even more true for the algae. They live in the water, they're out of our daily view. Things that happen out in the ocean far away, like I said in that poem a long time ago, is something that we don't really understand or relate to very well. I tried to make that more apparent to you today. So I hope that now you know that without algae, we would not be here. And energy flows, nutrient cycle. But, and I want to thank also several people, Bob Anderson and Lori Morris, that helped me with some information. John Ryder, who's the reason I came to Harbor Branch in the first place, some of the funding agencies. The work on the Inner Lagoon, some of it recently has been funded by the specialty license plate, Save Our Seas, and I appreciate anybody who's participated in that by buying a plate. But I think the last word really belongs to Einstein, And I think this time, instead of Walter Matthau Einstein, let's go with the real Einstein, Albert. And I said earlier, he has incredible commentary on a lot of things, and I like this one the best. If we knew what it was we were doing, it would not be called research, would it? Albert Einstein. Yeah. Thank you. Sorry about the technology problem with the program, but I think it was better to fix it. But I'll take a couple of questions. If anybody's got it. Otherwise, before you go, if you want, you can pick up one of these pieces of paper. You can help me decide my talk next year. All right, because I decided this is sort of late in the game. I came up with a title and I made the talk up. I can talk about deep water algae. I can talk about Indian River Lagoon, seagrass versus seaweed. I can talk about Indian River Lagoon water quality. I can talk about algae for a source of renewable energy. Something else, you tell me. Or I can take the year off. So if you want to vote, and anybody at 7 o'clock show gets twice the weighting of the people at the 4 o'clock show, because that's his practice. All right, anybody any questions?
We live on a lake in New Jersey.
You do?
Yeah. And
What part of New Jersey?
Highland Lakes, New Jersey, Vernon.
I know where that is.
Okay. We have a problem. Apparently it's not big enough for Rutgers to be interested in.
Ooh.
However, nobody can swim in that lake anymore. It is eutrophied. Now, we do have algae, and you can't catch it. It goes right through the nets. And
Yep.
so we have somebody who comes in and dumps chemicals, and
Yep.
it's good for one day, then you can't swim that day, and the next day you can't swim. So what do we do? Is there any way of harvesting this algae that could be beneficial, or getting rid of it so we can swim?
So her question was, we have a lake in New Jersey. It's got a lot of algae that's microalgae because they can't catch it with any kind of nets. What can they do about that? Can they kill it? They've tried, I guess, you said, with chemicals. Okay, getting back to my talk, nutrients recycle. So if you have an algal problem, you can't have an algal problem unless there's a lot of nutrients. And a lot of lakes, they're just collectors for everything that we do on land. It's like the lagoon, same comparison. Lakes usually don't have an outlet, or it has a fairly restricted outlet.
Right.
And so it has a high residency time. So those algae are likely to stay in that lake because they grow so fast. I showed you how fast they grow.
Mm-hmm.
If you kill them with a chemical, basically you're killing them, but the nutrients are still there. So those nutrients are just going to be recycled and fuel the next bloom of algae. So really the only way you can do that is to eliminate them, is to cut down any additional nutrients that go into the lake.
Can't be done.
Which you say cannot be done.
No.
But prudent land use sometimes can make an improvement, but it will take a very, very long time because there's so much nutrients in there. So you may very well be right. And I think it's a very hard one. I think the best thing to get rid of algae is actually filter feeders. And I don't know what we have some aquaculture people. I don't know what would grow in a lake in New Jersey. But zebra mussels would not be the right answer. They might grow in New Jersey, but yeah, that's honestly a very, very tough problem. The best you can do is to harvest the algae, and it's very inefficient. The only thing that harvests this microalgae efficiently is a filter feeder. It's very expensive to harvest them mechanically. I mean, it's basically like taking a pool filter and filtering your whole lake, which is not practical.
What is this other thing that you can do?
A filter feeder, an animal, a mollusk, and they live on the phytoplankton.
And they die too.
Yeah, they die. Everything dies in New Jersey. That's a bad take-home message. Anybody going to New Jersey soon?
So there's no solution.
I won't say there's no solution without knowing more, but it's not an easy one to solve. You have to reduce the nutrients.
Well, the houses are built along the edge of the lake, and at the time they were built, all the waste is going in.
Yeah.
A great deal of the waste is going into the lake.
Yeah. Yeah, we can talk more afterwards, but the answer is you've got to slowly reduce the nutrient inputs. You can do that by reducing certain types of land use. You can put aquatic macrophytes around the edge of the lake that will absorb a lot of nutrients that would be going in. But the problem is there's a lot of nutrients in there. If it's soft sediments, sometimes people will go in and actually harvest sediments out because a lot of those nutrients are tied up in soft sediments. They're doing that in the Indian River Lagoon. There's some muck removal projects that are going in, and the amount of nutrients that can reach suspended from muck is much, much higher than what it would be over like a sand bottom. So there are some things you can do, but I mean, none of them are quick fixes, and I'd have to know a lot more about the specifics. But we can talk more later. Yeah.
You showed a picture of a possible field of algae gathering in the sun's rays for energy use. Can you give a relative efficiency between using the sun's rays to raise the algae as opposed to having a solar panels like we see in use today? Is there a growth area in one or the other that's more efficient for the long run?
I don't know enough about solar panels to know the efficiency. Do you know what that is? That number is?
I know about 90% of the energy is still lost in basic heat loss. They don't have a good way to
The best algae could ever do is about 10%. So if he's right, then it would be very close. Algae hardly ever get to 10%. A much lower percentage of energy conversion occurs. A lot of the microalgal intensive cultures, some of the ones I showed you, they're trying to totally maximize light capture. And so you can push that number to 10%, maybe 12%. Some people think it go higher. But in our work, it was usually less than 10%. But that's high for plants. In natural environments, it's much lower. So, yeah.
Copper sulfate. What are the pros and cons of using that to kill algae?
So the question is copper sulfate. Pros and cons. Okay. The pros are, I guess it kills algae. The cons are, just like I was saying earlier, if it's ultimately a nutrient problem, you're just killing them in place. It's like one of my pet peeves locally is you ever see them remove aquatic weeds from choking up the canals, and then they just leave them on the bank, and then they decompose, and then they rot, and the nutrients go back in, and then the plants grow back up. Nutrient cycle. Remember, I keep trying to push that. So that's the problem. And copper sulfate, of course, copper is toxic to other organisms as well. So it's not a terribly benign thing to do anymore. Again, over the long term, these resources can be managed, I think, a little more efficiently, but it takes a whole change in our mindset, and it takes a lot of probably new students who come in and learn it that way, and that's what they expect will happen, moving forward.
Has any agency approached you with your digestion process that you had that produced a methane as a byproduct to pick up where you left off?
Any agencies ever contacted me about any of that? Yeah, I get called a lot. I get a lot of calls. Every time the price of oil goes up, it's going to go over $3 per gallon for gas, so I get called in. Yeah, I do. I'm involved in some small proposals that involve seaweeds. I don't really get involved with the microalgae stuff very much because that's not really my primary interest in, because fundamentally I think macroalgae will work better in the long run. So, yeah.
I guess if you'll indulge me, on the one topic, I'd love to see if you can do a prototype where you have some small water craft that goes through your nutrient process, creates methane, fuels this craft, but does it on a sustainable basis, where you let it loose to the Indian River Lagoon as an automaton, it goes for 10 years. But something to that effect to show the potential as a renewable source for maybe sustainable sea-based systems.
Yeah, I think that's an interesting idea.
And the other way I was wondering, because I oftentimes forget just the incredible amount of biomass that you talk about here. And some people propose that there's a Gaia hypothesis. They feel that when greenhouse gases get a little too far in extreme, the Earth reacts somehow to pull things back within the realm of you might call normality, or I don't know, equilibrium, I think is a better term. And I'm wondering if the biomass of algae has like these Gaia effects. In other words, as greenhouse gases go up and up and up, maybe we see baseline levels of algae increase.
That's actually a complicated question.
Yeah, it's not a fair question.
No, it's a good question. It's a complicated question because there's a lot of pieces to it. One of the things he asked about was the Gaia hypothesis, which basically says that the Earth, so to speak, will return to equilibrium. Is anybody in the room a Gaia expert who can answer that question? I'm not a big believer in that
Some people have
hypothesis.
things in equilibrium though, they exist in
Yeah,
thermodynamics.
I mean, eventually through time, again, we're talking geological time. I mean, what I did say is, the algae, have been here a long time.
Right.
They're going to be here a lot longer than our species is going to be here. That's not a doom and gloom thing. We might be here a long time. We probably will be. But the algae will continue to evolve. The algae will always be successful.
Yeah, those timetables,
So-
they could be Martians, right?
Yeah.
I mean.
And so, when you talk about the Earth's equilibrium and so on, I believe that's probably true over the long term. And we've had in past geological times, we've had carbon dioxide levels this high, we've had temperatures this high, we've had times when it's a lot colder. You saw the change in oxygen through time. That's been fairly stable for 500 million years.
Some people have a catastrophe theory where an asteroid hits every so often. It's hit Venus, they said. Earth a few different times, we've gone through cycles.
Yeah. So I don't know, does anybody else want to answer that? I'm happy to get anybody's other input on that. Go ahead. Yep.
If you wanted to do this thing on a national or even a global scale, working with microalgae or macroalgae as a renewable resource, as a biofuel, would you need to have different species for different climates and areas? If so, are there enough potential candidates?
That's an excellent question. I'm going to answer it more than what you asked. She wanted to know if we were to do like seaweeds or any algae throughout the world globally, would it be the same species or would it be different species adapted to local environments? The answer is it would be different species adapted to local environments. A lot of people who do seaweed and other aquaculture, the first thing people tend to do is they want to import it from some other place where it's been successful. For algae, I don't agree with that. There's almost always in the local environment, there will be a species that is incredibly adapted to that location. The best seaweeds that we have here in our system, which uses Indian River Lagoon water, what a shock, grow in the Indian River Lagoon quite well. And there's a lot of people who will want to take our species and put them somewhere else in the world, and I can name several countries that has been asked about. And I don't think that's a good idea. I think what I always recommend is a local screening program where one goes out and isolates the local algae and select what's most appropriate for the system. And the second reason I don't like that is because I mentioned some of the exotic species, invasive species. I mentioned the coating that I worked on a long time ago, and Brian LaPointe's work on chlorophyll. And a lot of the algae, can be a problem when they're introduced to another area. So basically, I think moving organisms around when it's not necessary is not a good idea. And there's always going to be exceptions. You got to remember, most all our agriculture is based on things that have been introduced from other places. So it's just, I don't think we need to do that. And I think you're better off using local species because they're going to be acclimated to the local environment. Okay, so good question. Yeah.
Well, wherever you go, I mean, we are basically the pollution problem with our animals and with our habitations. I mean, the number of people in the world has doubled since I've been alive, if not more. And we won't spend the money to really deal with it. I mean, her problem could be solved to put a deep sewer system around the lake, but somebody's going to pay for that.
Yeah.
A lot of people don't want to do it.
He's basically saying, he's been around for a while and population's probably doubled over his lifetime, and we have all these problems. And the only thing I'd say is, I think that I'm not sure the magnitude of our problems has actually increased totally as fast. I mean, obviously people is a problem, but I don't know that it's increased as bad as we might have thought. For example, in the Indian River Lagoon, we used to pump raw sewage out until mid-1990s. I personally feel like one of the best things that ever happened to the lagoon was that stopped. And I don't think we have enough information about how effective that was. I've tried to look at that a little bit, but not too seriously yet. But I think that would be a very interesting question on a local scale. There are cases throughout the world where, in freshwater systems and also in some coral reef environments, where, simply replumbing, the waste of humans or of the animals that are associated with humans has resulted in significant environmental improvement. So I tend to be optimistic. I tend to think that there's a lot. I work with a lot of students here, and they're very interested. The new generation of scientists, they're not so interested in saying, that's the problem. That's what my generation did a lot, baby boomers. We used up all the oil. That's the problem. I think the law students now, they're much more interested in conservation. They're much more into solving the problems. They don't go out and say, hey, there's another coral reef that's dying. They try to say, here's what we can do to make it better. I think you'll hear a little bit of that next week, actually, from Sarah. Some of the things that we're trying to do in that program.
I was gonna say that there is a new fuel cell that's been invented by somebody that uses a whole lot less fuel than anything. They're saying probably within 10 or 15 years, something the size of a filing cabinet, I guess, for want of a better term, will be powering an entire house for very little cost other than the initial startup to get them made.
He's talking about fuel cells. There's a tremendous amount, I mentioned the amount of money going in the biomass for algae. There's a tremendous amount of money that's going into fuel cells. And I'm sure there will be much better fuel cells. I'm sure there will be much better batteries. I'm sure that eventually we'll all be connected more. Our cars will be more electrical based, where it's part of the grid more. Where people who decide to be self-sufficient in their houses by solar, you'll be selling your power to the power company. You'll be getting money for it. Once the financial incentives are there, and they're getting there, I think you're going to see a whole new kind. If you've never read the book, what's it called, Flat, Hot, and Crowded by Thomas Friedman, you need to read that book. It's a very good book. Flat, Hot, and Crowded is the world's flat now because of the internet. We're all in the same playing field. If you can get on the internet, you got all the information at your fingertips. Hot, climate change, global warming, but as you know, climate change is more than just getting hot. It's getting erratic climate. And crowded is the population, which you talked about earlier, the number of people. That's a very good book, if you want to read that. That tells you about what the future's going to be.
You talked about the greater amount of algal use for food in, say, the Pacific countries. Do you see that as an increasing worldwide situation? Would there be an increase maybe in this country or Europe or other places where that could become something that could maybe solve some of the food issues?
He's asking about whether, the feeding habits, some parts of the world that eat seaweeds a lot, might we be able to take up and do that more? I don't know. I don't really like to eat seaweeds myself. It's like getting to work. I don't know. A long time ago, I thought that. I thought that would be like a new wave. I mean, one time, a bunch of years ago, Mexican food was kind of novel, and it really took off. And there's a number of other foods that have taken off, kind of. I don't know about seaweeds. What do you guys think? Seaweeds?
Oh, it's delicious.
Seaweed salad?
It's delicious if you go to a
Maybe I should. My next career should be on seaweed. Huh? What's that? What's that? Soylent Green. There you go. Yeah.
That was more than seaweed.
Yeah.
No, Soylent Green, remember what originally was the microalgae of the world. If you go back, Soylent Green, Soylent Red, and Soylent Yellow, they were the three, instead of brown, were the microalgae. That's what they were. And we use those up too.
So then we had to take a step up. We had to change the food chain.
Soylent Green.
So I'd like to think so. I mean, that's part of this whole thing is we all need to be eating lower on the food chain. The vegetarian diet really is much better ecologically. There's no question about it. And so I think you might see that. I don't know. I think it's going to take a while. I don't know, anybody else want to comment on that? I mean, that's just my own opinion. That's right outside my area.
I love seaweed, I love vegetables.
Now, Sarah can answer this question very good. She's very much a vegetarian, right? So, and she's healthy. Sorry, Edie, I didn't see your hand up.
Are you familiar with the Florida DEP Basic Management Action Plan for the IRL? That their endpoint is that they want the water quality to improve to the point where the seagrass beds are the same as they were in the 40s and 50s.
Yeah.
And so they've done all these efforts to reduce nitrogen, phosphorus, and I think in your region, the IRL is something pretty insane, like reducing it 60%.
Yeah, it's pretty high number.
50% for nitrogen.
Yeah.
But even if they do that, because nutrients are recycled, suppose they do that, do you think that that's
Okay, so Edie asked a really good question. It's really got two parts, I think. One is she's talking about the state, and they were actually kind of forced to do this by the EPA. We now are, for all of the freshwater areas in the Florida and all the estuarine areas, we're setting standards for how much nutrients we can discharge into those bodies of water. So sort of like your case up north. So I don't know if there's guidelines for it. But all states eventually are going to have to do this, so maybe that's going to be the solution for your problem. There actually, I don't know, do you know there's a meeting here on the 31st? Numerical standards, and it's going to be the DEP coming in and going through the calculations that Edie's talked about. But for like these places in Vero Beach, I think you're right. It's on the order of 60 or 70% of the nitrogen and phosphorus loading needs to be cut out. Now there are a couple things that are being done now to reduce the nutrient loading, and some of that involves cleaning the water before it goes into the lagoon. Some of this is as simple as catching the large plant debris that comes in. And others are to circulate some of the water through marshes, some of which are being man-made now in Vero Beach, before the water comes back out to the estuary. And some of it is to send the water basically further west and get it down flowing through the center of the state, because basically all this freshwater discharge only has happened in the last 100 years.